Okay, so another biological molecule that we need to know about is lipids or fats. That's the same thing. So our most common lipid or fat would be a triglyceride. So now a triglyceride is made up of 3 fatty acids and 1 glycerol molecule. So now a fatty acid contains a carboxylic acid group and hydrocarbons. So a carboxylic acid group is carbon that's double bonded to an oxygen and an OH. And then a hydrocarbon is just a long chain of carbons attached to hydrogens. So this carbon chain can have anywhere from a few carbons all the way up to 18, 20. It can just keep going with that. If this chain is single bonded, we call it saturated. So that has single bonds. If there is any of those carbons that is double bonded, any one that has 2 bonds, it's unsaturated. So monounsaturated has 1 single bond, polyunsaturated has double bonds. So anytime we have a monounsaturated means 1 double bond, many double bonds is polyunsaturated. So now in this chain, if it is saturated, we usually represent the chain without the carbons and the hydrogens, and you represent them by just points. So that is carbon with my carboxylic acid group, and then the rest of the chain, each point is a carbon on there. So anytime you see this zigzag shape, that represents each carbon, and that's my long carbon chain. As soon as I have a double bond in there, what happens is, where the double bond is, the chain becomes straight. And then you have the rest of the chain. So you end up with what we call a kink in the chain, because a double bond would cause it to change shape because it ends up being straight. Whereas this is just a zigzag. Anytime you see that, it means that there was a double bond. And it could cause it to change direction, facing more downwards, facing more upwards, something like that. So now in these triglycerides, there's the fatty acids, which look like that. But then there's also a glycerol. And a glycerol molecule has alcohol groups. So it is 3 carbons, and each one of those carbons has an OH on it. And then the rest of the bonds around there are hydrogens. So that is a glycerol because there's 3 carbons with 3 OH groups. Okay, so we have a glycerol combined with 3 fatty acids. So when we have that, the glycerol can line up with a fatty acid, and on that carboxylic acid group, the OHs line up. And when they line up, it means that those molecules can come off as water, and the oxygen bonds one chain to the next. Same thing can happen with the next fatty acid chain and the last one. So water can come off 3 times, and then it makes 3 bonds. So this molecule would be carbon, carbon, carbon. The H's around the outside remain the same, but now they're bonded in the middle by oxygen. To my other chain on the other side, which still has its carbon double bonded to its oxygen and its long chain. Okay, and remember that can be any number of carbons. So now this bond between where we had our glycerol and our fatty acid is now called an ester bond. So in carbohydrates, we had glycosidic bonds. In fats, we have ester bonds. And it makes 3 water molecules at the end. And those molecules we call metabolic water. So when we join lots of fats together, we make 3 metabolic waters, which is more than any others. And that's why, also because there's so many bonds in here between the carbon and hydrogen in that long chain. That's what stores all the energy. And that's why fats are a good storage form of energy, because there's energy in that long hydrocarbon chain. So that's why our bodies prefer to store things as fats, because then it gives us lots of energy reserves, which is the main function of a fat, as well as for insulation. Okay, so we have 3 ester bonds and 3 metabolic waters. And when we go this way and we make water, just like before, that's called a condensation reaction. And if we're going to break them and add water and break all 3 of those bonds, it's going to be hydrolysis. So that is then our triglyceride. The other fat molecule that we have that's special is called a phospholipid. So now a phospholipid is the same as a triglyceride, but we've replaced one fatty acid chain with a phosphate group. So we're still going to have our glycerol molecule with its bonds around, the ester bonds to the fatty acid chain, but one of them, instead of being that fatty acid, it forms a phosphate group. And that phosphate group is special because a phosphate likes water, whereas a fatty acid does not. So we can simplify that by just representing our phosphate head and 2 fatty acid tails. So now the phosphate we say is the head, and because it likes water, we say that it is hydrophilic. Because hydro means water, philic means love. So it loves water and it is water soluble. But these 2 fatty acid chains don't like water. So we call those the fatty acid tails. And we say that they are hydrophobic, meaning water and fear. So they don't like water. So now a phospholipid molecule has both a hydrophilic part and a hydrophobic part. So that plays a role in their arrangement around, because that part is water soluble and that part is water insoluble. So when we look at our cell membrane, that's going to become important. But for now, this is all you need to know about the structure of fats.